All right, hey folks, welcome to another edition of Zog Science. Today we're going to be talking about kind of an introduction to cells and the endomembrane system. So when we're talking about cells, we need to figure out how to actually study them, and we do that using microscopes. The reason why, cells are pretty small, and so there are kind of two different cell types of microscopes that we can use, either light microscopes for low resolutions, or electron microscopes. We will not be using electron microscopes in school because those are pretty expensive and sophisticated pieces of equipment. We will instead be using light microscopes. The reason why cells are small is because of the fact of the surface area and volume. Cells need to get things in and out of themselves rather quickly in order to be able to live. So the smaller a cell is, the higher its surface area to volume ratio is, and therefore it can move materials in and out more efficiently. Larger cells have a smaller surface area to volume ratio, and therefore they have a hard time getting rid of waste. So there's actually a physical restriction on how large a cell can get before it is unable to get rid of its waste and will need to go through mitosis, or it could die. Cell fractionation is basically centrifuging. It's a way to kind of separate out the parts of a cell by the mass. So you take cells, you kind of kind of mix them all up and blend them, and you get a homogeneous or kind of a uniform test tube full of cell. You then centrifuge the cells, so you spin it. When you do that, all the heaviest things will go to the bottom and form a pellet. You can then pour off the extra of the supernatant, which is the liquid above the pellet. Then you take that supernatant, you spin it again at a higher speed for longer. You get again, a pellet that forms at the bottom with more parts, and you continue to do that until you have separated out all of the cell into different parts. And you can kind of see that in this picture here, all the different things that you can separate the cell into. We will do some centrifuging, not of cells, but we will do some centrifuging during class. So the life of a cell, what are the things that a cell needs to do? Well, it needs to be able to process matter, so it needs to be able to pull in food particles, essentially, and be able to synthesize and digest those food particles. It needs to process energy, so it needs energy to be able to do that. And then it needs to process the information found in its DNA or information that is coming from outside. Some cells also reproduce, so they need to be able to pass on the information. And then some cells, they need to be able to communicate. So they have to be able to talk with other cells, especially if they are multicellular, but also unicellular cells, organisms communicate as well. So some common features of all cells, there's the cytosol, which is essentially kind of like the cytoplasm, but it's the actual stuff that's inside the cytoplasm. It's a semi-fluid, and it has all of the subcellular components, so all the things that are kind of smaller than a cell, those are all suspended inside it. We've got chromosomes, which are the structural form of DNA. Ribosomes, which produce proteins using the DNA instructions, and the plasma membrane. Those are things that we find in all cells. So we kind of classify cells into two main parts. We've got prokaryotes and eukaryotes. Prokaryotes are generally smaller, less complex, and they don't have any membrane-bound organelles, and they're much more abundant. There are way more prokaryotic cells in the world than there are eukaryotic cells, partially because they're so much smaller. Eukaryotes are generally larger, more complex, with lots of membrane-bound organelles, and they are less abundant, much less of them. You can kind of see over here a little bit of a comparison between a bacteria and then an animal cell and a virus. You can kind of see the size differences. So why do you think membrane-bound organelles might be important? Well, think about organelles like compartments of a submarine, okay? And so having different compartments in a submarine allows the different parts of a submarine to function effectively, function efficiently. So an organelle allows the cell to have separate sections within itself, and membrane-bound organelles in particular. So the membranes kind of separate that environment of the organelle from the rest of the cell, allowing it to be able to function at a different pH range, have different salts, have different molecules inside of it. And so that allows it to really carry out its function much more efficiently than if it was just kind of in the cytosol, in the cytoplasm area. So that specialization is really important, and it also allows cells to become larger and be able to do more complex things, and eventually leads to multicellular life. So the endomembrane system, basically is a lot of the organelles that are going to be involved in the jobs of protein synthesis, transporting proteins to membranes and out of cells, as well as some of metabolism, although most of metabolism is done by some other organelles that we'll learn next time. And then moving of lipids and detoxification of poisons. And these are some of the organelles that are found within that system. So we've got the nucleus. It protects the DNA with a nuclear envelope, which is a lipid bilayer, so very similar to the membrane that you find on the outside of the cell as a whole. And the nuclear envelope has pores in it that allow some molecules to pass through. DNA is not able to pass through because DNA is too large. However, proteins and RNA are able to pass through those pores. And you can kind of see what the pores look like over here underneath an electron microscope. And then we've got ribosomes. Ribosomes are the protein and RNA structure, and they consist of a large and a small subunit. There are kind of two types: free ribosomes, which float around in the cytosol, and then bound proteins, which are attached to the rough endoplasmic reticulum. Free-floating ribosomes produce proteins that are used within the cell, and bound ribosomes are the ones that are found in the rough ER, and they make extracellular proteins, or proteins that are going to be sent out. So kind of the main kind of deal with the endomembrane system is that we're starting with the nucleus, okay? We've got the information that's found inside the nucleus. That information is then going to be sent to the rough endoplasmic reticulum, or ribosomes, free-floating ribosomes. But in particular, the endomembrane system, we're focusing on the information that's going to the endoplasmic reticulum. And it's because it is located directly next to the nuclear envelope. So the endoplasmic reticulum kind of surrounds the nucleus, and you've got both the smooth and the rough endoplasmic reticulum found right there. You can kind of see that there you've got channels, and how it would be very easy for the RNA that is leaving the nucleus from the process of transcription to come out into the rough ER and do its job. So there are, again, two types of rough or of endoplasmic reticulum. It's kind of these folded membranes, and they call them cisternae. You don't need to know that necessarily, but it is important to kind of, be able to recognize the structure. The smooth ER synthesizes lipids. It helps to metabolize carbohydrates, detoxifies some drugs and poisons, and it stores calcium in the cell. It doesn't have any ribosomes on it, which is the reason why they call it the smooth ER. So you can kind of see what that looks like here in this picture. The rough endoplasmic reticulum, you can see, has little bumps on it. Those are the ribosomes. The smooth endoplasmic reticulum obviously doesn't. The rough endoplasmic reticulum is studied with ribosomes, so therefore, based on that, you can kind of assume that its function is to synthesize proteins, and you would be correct. And in general, it's going to be synthesizing proteins that are going to be sent out of the cell. So what happens is that the proteins are synthesized, and then they erupt in a membrane called a vesicle, which again is just a lipid bilayer, where it is going to be transported to the Golgi apparatus. The Golgi apparatus is kind of, a lot of times it's likened to the post office. So what it does is it takes vesicles from other proteins that have proteins or other molecules that have been synthesized in other places, in general from the rough ER, but they can be synthesized elsewhere. And then they kind of modify the proteins, the packaging, and then they ship it out. So they kind of receive these proteins, they might change them a little bit, they put them in a new coating, and then they ship them out. And in general, what happens is that those kind of vesicles that are sent out from the Golgi apparatus then get sent to the cell membrane, where they are then the proteins are then released out into the extracellular space. We also have some other membrane-bound organelles that are kind of a part of this system. You've got lysosomes, and lysosomes contain hydrolytic enzymes, so it just means that they use water or hydrogen peroxide or something like that to be basically digesting macromolecules. So a lot of times what happens is that food is brought into the cell through this process of phagocytosis, where the cell membrane kind of pinches and brings the food in. A lysozyme will then come and merge with that vesicle or food vacuole at that point, and then release enzymes inside, which are going to be breaking down the food particles. Lysozymes also function in the digestion of non-functioning other organelles. So, for example, if mitochondria is damaged, lysosomes can then bring in their enzymes, release the enzymes inside, and break it down and digest it so that it can be recycled. So they're really useful in terms of helping cells survive and use their functions. Vacuoles are kind of large vesicles, and sometimes they can be considered lysosomes. It just kind of depends on who you're talking to, but in general, we're going to think of them as something different. And there are three different kinds. Food vacuoles, which is kind of talked about, they formed by phagocytosis, where the outer membrane kind of pinches in, it brings food into the cell. Contractile vacuoles help pump excess water out of the protist cells, and generally these are going to be freshwater protists, helping maintain homeostasis, so they maintain the right balance. And then the central vacuole, which you all have learned about in biology before, is found in plants, and it stores a large amount of water along with some inorganic ions, so things like potassium that they need to survive. So here's kind of a wrap-up of the endomembrane system. We've got the nucleus sending out things to the rough ER, which is then going to, through vesicles, send to the Golgi apparatus. The Golgi apparatus then sends vesicles to the plasma membrane, which is then going to be released out into the extracellular space. And that's all I've got for you today. Thanks for watching, and I'll see you next time.